Well, first off, my name is Giulia. I'm the application engineer at CryoSol, and I'm working on the development of the VitroJet. I'm very excited to kick off this morning's session. I think you'll see a lot of cool videos coming up from all the speakers. So to start with, as we've already heard even in the previous talks, sample prep is becoming a hot topic and a huge issue in cryo-EM, especially in single particle cryo-EM. So let me just start with an overview of the general workflow. So if we start with the sample all the way to your reconstruction, we've seen that a lot of advancements have been seen in the microscopes. We have more powerful microscopes, automated acquisitions, better detectors, new softwares to as accurately as possible, reconstruct your three-dimensional molecule. And that's great, but that's really shifting the bottleneck more than ever back to the sample preparation part of it. And that's really what we focus on in the VitroJet development. So from depositing a desired thin layer to freezing it, and then for the microscope. So I used to have a slide just showing the workflow of grid preparation. I took that out. I think you're all very familiar with it, but it's pretty clear that it's tedious work. There are a lot of user handling steps and a lot of places where problems can occur. And I think you'll agree that one of the biggest issues is the reproducibility of the grid preparation. And that's what we kept in mind in the development of the VitroJet. So we wanted to have basically an integrated and automated system. That means your sample carriers, as they'll go into the microscope, so pre-clipped in the autogrid form. These are placed in a supply cassette. We have an integrated plasma treater so that we have reproducible wetting conditions for each cycle, each vitrification cycle. We use a blotless deposition technique called pin printing, which I'll describe a little bit more in the upcoming slides. And what allows us to vitrify the autogrid is the use of jet vitrification. So we start from targeting from within the sample outwards. And finally, these prepared grids, are stored in a grid box, and the user can choose to either go to the microscope or store them for a later use. Of course, we wouldn't have done this if we didn't foresee the benefits in the process. By automating the process, you reduce the inter-operator variability and the necessary training. You start with a sturdier sample carrier, so prevent the grid damage, and by removing the extra handling steps, we hope to reduce contamination. Our technique only starts with half a microliter of stock volumes, and then you can basically make as many grids as you like from that. And what I find most important right now, especially because we're still working with a prototype model, is this visual quality inspection. So we actually see exactly what's taking place in the deposition, and we're trying to take advantage of that as much as possible. I think the upcoming slide. So recently, we used to just show the animation, the 3D animation, which really shows the process, but we recently had the process filmed and mastered, so you'll see the instrument as it stands now and what the workflow looks like. And you'll see me as the star of the video, so just keep that in mind. So this is our machine, still shiny with a bunch of wires and stuff coming out from different places. And we start with the pre-clipped grids in the supply cassette. And the supply cassette is placed within the device. Then the sample is taken up. We have this automated pipette, and half a microliter of sample is taken up, and it's placed directly into the climate chamber. And then the entire process begins with the grid. So there I am looking a bit confused. The process starts. So grid is picked up. First step is the integrated glow discharger. So here you can tune the parameters as you like, and we are looking into different plasma treaters currently. And following that, the grid is taken out, and then you go to the deposition chamber. So just something to keep in mind in this video, because this is actually what we see whenever we work with it. On the bottom screen, so you see two screens. Eventually it will be reduced to one. But on the bottom screen, we monitor the conditions and we set the desired conditions for the deposition chamber. And on the top one, we can see what's taking place. So you see some movement going on in the machine. Next, you'll see the focusing of the pin. So we use a solid pin, and then the focusing of the grid. And that's what we see from our camera behind the grid. Then you see the pickup of the droplet and the deposition. And just a close-up example of what that looks like. Something to note here, this is one of the earlier videos. We used very minimal grid coverage. Now we fill at least the entire field of view. And of course, we are limited by that field of view, so we are upgrading to a better camera. But that's what we see. And the last step of the process, it used to be a slow motion video, so chances are you might not really catch the freezing, so just keep an eye out. Basically, your gripper is plunged down and it's quickly frozen by 2 jets, and then finally the grid is stored in the grid box. And when that's all done, we use the same tools as FEI to close the grid box, and then you can do whatever you like with your grids. Okay, so I ran you through the process. Now, to understand what we use for deposition, recently we've looked more and more into understanding this process. So starting with how the droplet forms, the droplet pickup. Like I mentioned, we use a custom-made pin. So we have to look at the droplet formation on the tip of this pin, because that's what you'll have to spread across your grid, to deposit on your grid. Then we looked at deposition. So this is a technique that offers a lot of parameters. We can play with the pin parameters, so we can start with bigger pins, smaller pins, if we want to cover more grid surface, and different cone angles will also leave you with a starting residual volume, a different starting residual volume. Of course, depending on what's in your buffer, you can alter your velocity of the pin being dragged across the grid. The pin never touches the grid. It's maintained at a standoff distance, and that can be tuned too. That's why we put both components into focus first. So that's being studied more and more, also to take full advantage of it. We took into account the EM grid properties, because by introducing the grid bars, there is some natural wicking that takes place, and any evaporation that occurs. Of course, we try to minimize that as much as possible by maintaining the sample at its dew point. But it's very difficult to really accurately maintain it. I think you'll always either tend to go towards condensation or evaporation, so that's taken into account. And finally, we're working on a model to be able to take full advantage of the deposition. So that means if we can predict, already we know whether the deposition that took place will be suitable for the microscope. But this is from a few expert users. So we wanted to develop a model to predict the whole thickness. This is just to show some examples of what I mean about seeing the layer as it's being deposited. When there's evaporation taking place, you can already see the holes opening up. I'm not sure if you can see it on this screen, usually if it's a little bit darker, but you see the holes are open here. So that's an example of something that you wouldn't put in the microscope. This is a desired layer thickness, because you see that it's based on the intensity, it's a thin layer, and the layer is maintained. There's no evaporation taking place. Then when you start playing with the parameters, you can make the layer thickness larger, and this would probably look like a black grid in the microscope, at least the bottom corner. These are just some initial results. We are trying to use the intensity filter. So this was a collaboration we had with Groningen University. Of course, we're not there yet. This isn't the final correlation, because as you can see there, we are off by about 20 nanometers for the low range. We do see some nice correlation for the higher values. But on the left, you see the image that's taken from the VitroJet. So we looked at the intensity difference from before deposition and after deposition, and then looking at the measured values from the electron microscope, we found a correlation between the intensity and those values. And these look like pieces from a Lego or something. But these are actually some of the features that will be coming up. What I didn't mention yet, because most of our time we're spending on developing, well, getting more understanding in our processes. But we are also developing beta models, and these will be coming out late this year and early into the next year. Hopefully earlier rather than later. But these are just a couple of the features that will be implemented. So first, autofill will be maintaining our levels of liquid nitrogen as well as liquid ethane throughout the process. So that's why it's a larger cryogenic container. You'll start with basically a stock volume of your liquid nitrogen, and that will be maintained throughout the process. And we will have a new climate chamber. So this is really the key to our process, to maintain the dew point on our sample, to really be able to predict and deposit layers as we want them to be, so that the user will have actually the desired layer thickness that they're looking for. And with that, I think I'll conclude. And just a few people to acknowledge. So we are part of a spin-off company. We're working very closely with the University of Maastricht. So on the top, you'll see Peter Peters' group with Raymond and Carmen, who run the lab. And we work very closely with them. On the bottom, you see again the spin-off company, along with a lot of the engineers who are working hard to develop these beta models. Or they want us to call it first-generation VitroJet models, but, you know, same thing. But, yeah, thank you.
Thank you very much. Questions?
Yeah, so we are working with a number of different grids. We've tried C-flat, the quantifoil copper, gold, I think in the different hole sizes, and then graphene grids and so on. And I think for each grid you have to take into account the relative dew point of the grid, because, so that's where I think you see the most difference. You have to look at the evaporation that can take place and take that into account. This is just the feeling I sometimes get. Sometimes we get better, thinner layers with the gold grids, but that's maybe that's just a personal thing. But yeah.
How much time between the deposition and the plunging?
Yeah, so that depends. We can speed up the deposition, but it is a slow process. I mean, if you're comparing it to, I think, some of the other presentations, so we are in the order of a couple of seconds, and then the process of bringing it down to the cryogenic chamber is really quick. Really quick for me is 80 milliseconds. So, yeah.
There seems to be a lot of evaporation.
Hi.
Seems to be a lot of evaporation during the grid thinning. So have you looked at how that might affect salt concentrations and buffers?
Yeah, well, we do try to minimize that as much as possible. So that's the whole scope behind really working on our pinprint models, is because we don't actually want the evaporation to take place, and we do consider that as harmful for the protein or whatever molecule you have in the liquid. So we do try to minimize that. So we haven't really looked at the effect of the evaporation on the sample.
Yeah, I think that could be important for some samples.
Sorry?
It could be very important for some samples.
Yeah.
Okay, I think we have time for one more question.
Do you know if the jet dislodge more layer of cells?
Sorry?
Do you know if the jet dislodge more layers of the cells?
No, well, we haven't worked with cells yet. We primarily worked with proteins or some larger components too of the cell, but the vitrification is so quick. So I think that's a common misconception, that by jetting you'll destroy the grid, but actually the vitrification takes place in 1 millisecond. So by the time the stream actually hits your grid, it's solid. So we haven't had any.
Well, I think it's based on the jet propane concept.
Mm-hmm.
I remember that the jet propane concept dislodged the
cells.
layers. Have you tested?
Well, we have also another machine that we use for some cell vitrification, but there's no, I don't think I haven't quite worked with that. It's something that's in-house, but I don't think we've seen any dislodging of cells. So, but it is really important. Just something to point out. It is very important for the jets to hit simultaneously. So maybe if you're off, then it could take place.
Okay, I think in the interest of time we should move on. Let's thank Giulia once again. Thank you very much.